Doing an audio check here. All right, audio check. Looks like we're good. Yeah, I'll be definitely doing that at the beginning of all of these because an hour and a half goes by and I've recorded a whole lecture and it has no audio. That has happened a few times. And I think I'm actually going to, just thinking about, it actually just happened before I gave this lecture. So I might start doing them on PowerPoint. I think that makes a local copy. It's not, I don't know. Anyways, we're going to dive right here into chapter 1. So the other day I was talking about your exams, and generally the exams are, you have an even distribution of questions according to the chapter that they're coming from, right? So exam 2, which is going to cover chapters 4 and 5, it's a 40-question exam. Pretty spot on there. 20 questions are from chapter 4 and 20 questions are from chapter 5. Now, for this first unit exam, that's really the only one this semester where that's kind of an exception. The questions are a little light from this chapter in chapter 1. And that's because this chapter is very introductory. There's really little science that we're going to go into in here. It is it's an interesting chapter, though. I want you to read it in the book. I want you to answer the questions at the end of the chapter, but really all it covers is, why genetics is important. It goes through the history of genetics, which, some of these scientists you should know, but what I really want you to understand is kind of how the field evolved. We're going to talk about that a little bit. And then this chapter also outlines and presents to you some very fundamental terms and whatnot, and principles. All right, so why is genetics important to us? Well, think about it. Genetics defines who you are. It contributes to your personality. It contributes to, are you more likely to get this disease or not? Do you have a natural talent for baseball? It's very fundamental, not only to individuals, but also to ethnicities and populations. We are our genes. It's not everything, but it's a big part of who we are. Certain ethnicities have a predisposition to certain diseases. And those are all things that need to be taken into account. So very important aspect of biology. As an example, so this is a very specific gene, nice scientific name, SLC26A2, which, as we're going to learn later on, the nomenclature usually has a lot to do with the locus of the gene. The locus is the location on the chromosome. So here, this gene is located on chromosome 5, and it's the MUT version. So that's actually not pronounced MUT. That would be 2 T's, right? It's pronounced mute, and it's the mutant version of this gene. So when individuals have a mutant version of this gene, they are very susceptible to a disease called diatrophic dysplasia. And diatrophic dysplasia causes all these skeletal abnormalities. You get things like hitchhiker's thumb and abnormal growth of bones and like overgrowth of cartilage and then undergrowth in cartilage in some areas. Everything's all messed up. Now, just to give you guys a little something, I am not an expert on genetic diseases. It's just out of my realm of training. So, if you ask me what is this gene of this mutation, I might not know. I'm probably not going to know. I'm a real cell and molecular guy. But as we go through each of these chapters, there's going to be a good, disease case study presented in each chapter. And that you definitely want to learn these examples because these are the examples that are going to come up on your standardized exams, the most classical examples in genetics. So anyways, this dysplasia gene, you can see here's a normal skeletal hand individual that has dysplasia. And, that's caused by a mutation in one gene. That's all it takes. All right, so genetics is important in agriculture and biotechnology. This guy, Norman Borlaug, he basically developed these high-yielding wheat crops that have really, well, for a while, contributed to the ability to feed, such a great number of people on the planet. We got the Nobel Prize for that. And, genes are important in biotechnology, medicine, for providing things like insulin. That's the classic example. Provides antiviral drugs, antibiotics, enzymes, right? Vaccines. We're going to talk about a lot of examples of this in our course. And genes are important in the study of biology, right? If you're trying to characterize an organism, well, you want to look. First thing you want to do is sequence its genome. That's what we do now. And so genetic features, genes are highly shared among organisms. This kind of led the belief that, we all kind of arise from a single, in terms of evolution, a single organism, right? One cell. And over those millions of years, that has developed and expanded out into all the species that we know today. But evolutionary biologists, molecular evolutionary biologists, study those, how those genes backtrace to that single primordial organism, and also the homology among different organisms in terms of the genes that they share. All right, so just a term here, a genome. So a genome is the complete set of genetic instructions, right? So, your genome is all 46 of your chromosomes in your cell, and it has all of the instructions on how to make you, okay? And you'll find that instruction booklet in all of the cells in your body except the red blood cells and the gametes, right? So those are a different set of instructions, and we'll talk about that later on. But your skin cells and your liver cells have the same instructional blueprint. So genome, or that root of the word ohm, comes from omic, and that root word means global, right? So if you're doing a genomic study, what you're studying are all of the genes of the organism. Now, there are other fields of omic biology: proteomics, transcriptomics, metabolomics, right? So when you're doing a proteomic study, you're looking at all of the proteins in the organism or the cell. These omic fields have just blown up over the last 20 years. Proteomics now is really at the forefront of cell and molecular biology. And but genomics is catching up, and it's going to be, in the end, ultimately, once we have an AI system that's capable of interpreting a genome, that's when really there's going to be no jobs for biologists anymore. I'm just kidding. Okay. So now, when we talk about genomes, right, it can be either RNA or DNA, because not all living organisms, right, they have DNA, but viral systems have both RNA and DNA. In genetics, we talk about viral systems, even though they're not considered living. These genomes copy during the process of cell replication. So during mitosis, the two daughter cells have the whole genome that the parent cell had. And this genetic code, right, the code of DNA, the ATCG, it's the same in all living organisms. Right? That's kind of cool, because that enables us to transfer genes from one organism to another. You guys have ever seen a picture of like the pigs that have these glow-in-the-dark noses? Well, that's because a molecular biologist took a gene from the jellyfish and put it in the pig. And now, certain areas of their paws and their noses glow green because of that green fluorescent protein. Really cool saying. I've actually got this saying on my profile, my St. Pete College profile, that life is a river of DNA that runs through time. Right? This really connects all organisms, past and present. Right? This this magical system of encoding genes in nucleic acids that are just made up of 4 different chemicals has not changed in billions of years, right? Ever since that common ancestor, that primordial cell, gave rise to all the life that's on the planet. When I talk about this stuff, I always think Morgan Freeman could do a lot better job, but when we get into the nitty gritty, I got Morgan Freeman. Take him out. Okay. All right. So concept check here. See if you guys can come up with this. What are some of the implications of all organisms having similar genetic systems? Don't cheat. Don't go to the answer. Let's read these through. All life forms are genetically related. Are you related to a jellyfish? Well, that research findings on one organism's gene function can often be applied to other organisms. That genes from one organism can often exist and thrive in another organism. Well, that's true, but because I just mentioned an example of that. So what is the answer there? Well, the answer is, in fact, all of the above, right? All life forms are genetically related because we all come from a primordial ancestor. Don't tell your mom or dad that you're genetically related to the jellyfish. They're not going to understand that. But in the field of genetics, we say all life forms are genetically related. And genes from that research findings on one organism's gene function can often be applied to another organism's, right? That's definitely true. So, if you have the gene for tubulin that you're looking at in a C. elegans, which is a tiny roundworm, and you're comparing that tubulin gene to a gene in the wildebeest, and it's got the same sequence, or at least similar, well, then you can infer that the function is probably similar as well. Okay, so these are all implications of organisms having similar genetic systems. All right, so how is genetics divided up? Well, it's basically divided up into 3 fields: transmission genetics, molecular genetics, and population genetics. Now, in this course, we are going to talk about the first 2. And if you want to get into the third one, population genetics, you will cover that in ecology. You'll start to get into population genetics. You're going to talk about the Hardy-Weinberg principle and all that stuff. The very last chapter or 2 of this book has some inference in population genetics, but nowadays traditional general genetics really just focuses on transmission and molecular genetics. And that's because population genetics is like a whole other animal and requires some understanding of evolution. By the way, evolution, if you haven't taken it yet and you are a bio major, I highly suggest that you take it for 2 reasons. It's not a crazy difficult course. I guess that's going to depend on who you take it with. But it is also, and everybody's different, but it was definitely the most interesting course that I took in my undergrad. I loved it. And that was 30 years ago. So I'm sure it's gotten a lot better, since then. But even at the time, it's an amazing subject. And I really encourage you guys to take that course. I'm not sure. I'm pretty new here at St. Pete College. I don't know who teaches that here, but anyways. Okay, so now these 3 divisions overlap, right? Transmission genetics looks at, how genes distribute among progeny pretty much, like how they pass from generation to generation, whereas molecular genetics looks at, the DNA code and how that is expressed out in terms of proteins. And then population genetics looks more at, the genes that are common in certain populations and what's the degree of that commonality and whatnot. But all 3 of them do overlap. And this kind of diagram, this is a very common diagram that's used it's a common way of representing data that in omic biology, when you're looking at genomes, trying to compare genomes, or you're comparing transcriptomes, called the Venn diagram, when you've got these 3 overlapping circles. So transmission is what we're going to do the first half of the semester, and molecular pretty much the second half of the semester. All right, so transmission genetics. This is this is Mendelian genetics or classical genetics, as it's called. And these are the basic principles of how genes are passed from one generation to the next, which is called heredity. And one thing, how this differs from a big difference between this and population genetics is we're really focusing on a single organism, right? Not a group of organisms. Now, we may look at a pedigree, right? Which is a pedigree is a series of symbols, a figure that shows, the transmission of traits across different generations. But usually when you're solving a problem in that pedigree, you're focusing on one individual in the pedigree and you're trying to solve the genetics of that individual. And that's what's meant on the focus is on an individual organism. Molecular genetics, we're at the gene, the level of the gene, and how the genetic information is encoded, expressed, how it's copied, how maybe it moves about the genome. We'll talk about mutations. There's a lot of ties in this to cell biology, and that's why your success in this area of the course, will be a little contingent on the level of background that you have in biology. If you did well in Bio 1, not necessarily Bio 2, it's not as relevant, but if you did well in Bio 1, or if you've taken cell biology and you've done well in that, a lot of that stuff is overlaps, right? But generally, people have that background in this course, and it's really all the transmission genetics that's very new to them. And they find the first half of this course a little more challenging. So just my 2 cents based on my experience with students. All right, and population genetics, which we're not going to talk a lot about. We'll talk about a little bit maybe when we get into like bacterial systems, but this is the genetic composition of population. So how that composition changes over time and geographically, spatially, and with population genetics, you're focusing on usually more than one gene and obviously more than one organism. And of course, you're relating it to time, right? You're tying it to evolution. So population genetics and molecular genetics are definitely considered the more modern genetics, and it's a very broad field, right? It's there's more specifications, there's more specific fields, subfields within that currently than we're going over in this course. Okay, so if you want to study genetics, well, you need to use a model organism, right? And we can't study all genetics in people, because especially molecular genetics, because we can't give someone the gene for jellyfish. That would be ethically, and that would just be crazy. So using model genetic organisms is ideal. So we're going to talk about a lot of them in this course and give some very specific examples of them. I've got a list of them here. We're going to talk about Drosophila. That's big in transmission genetics. Thomas Hunt Morgan is like the father of Drosophila genetics. If you're taking the lab, we're going to do some pretty cool crosses with flies. You're going to count flies and mate flies and type flies. We'll be also working with E. coli in the lab and C. elegans as well. And if you look at all these organisms, the mouse, the yeast, Arabidopsis, C. elegans, you can start to think, well, the first thing is they're kind of small, right? So, we don't have a big organism here that we're using. And, well, why is that beneficial? Well, things that make, why is a model organism a model organism? These are the features of them, right? So short generation time, okay? So, they have their progeny, and we don't wait 10 years for them to grow up, and we can observe phenotypes, right? Either a number of hours or, weeks at the most, like for a mouse or Arabidopsis. But short generation time. Also, having numerous progeny is great, because now if you need to do statistics on, the percent of throughput that a certain phenotype has, then you have a lot more progeny to work with. Ability to carry out controlled genetic crosses. And this is why, it's one of the big reasons here why Gregor Mendel is the father of genetics, because he could actually do these like simple surgeries with his pea plant. He could do self-fertilizations and cross-pollinations very, very carefully and very easily, and be able to control the genetics of his model organism. Now, the pea plant isn't on there. These aren't all of the model organisms. They're the most commonly used today. And that's because the genetics of the pea plant has been pretty thoroughly investigated since Mendel did it in the 19th century. But it is still considered a model organism. So you want to be able to do this all in a lab environment, right? If you're doing you're doing people, can't do people in a lab environment, can't grow them in cages and stuff. And also, the ability to have numerous genetic variants is also very important, right? So, and you can run into this problem sometimes with certain viruses, but you have to have a good number of genes where you can easily detect the phenotype, right? If you have to do this complex molecular analysis to determine if the phenotype is on or off, that can make it more difficult. Doesn't mean we don't do it, but they need to be kind of, the easier to detect, the better, and there should be a lot of variants as well. And this is probably the reason why, you don't just grab a lizard from your backyard and start using this as a model organism, but having an accumulated body of knowledge that you can compare your results to and that you can apply to your own results is very important. So having the genome sequence for Drosophila is a great benefit, right, in your research, because you can do PCR and isolate a gene out of the Drosophila, and you can get the sequence, and then you can compare it to its genome. Right? A lot of people have already done that work for you, and you can compare your results. All right, so we're going to be talking about all of these, although I don't get in too many Arabidopsis, but we need to talk about a few. When we get to epistasis, I think was an example. All right, so here's an example. Zebrafish, great model organism, wonderful model organism, and not in this list right here. So there was a gene that was recognized in the zebrafish and showed a very close relationship to pigment variation, right? And unlikely that this gene, the purpose of this gene, would have been discovered by studying humans, right? Because of all the genetic manipulation that you can easily do to zebrafish. I worked in a zebrafish lab in grad school for a little while. It was great. We had a big fish room with all these fish in there. One thing about zebrafish, it's very easy to inject the embryos. And so if you're trying to make transgenic organisms, this is a great model organism to do it. And this discovery relied on that ability to be able to generate transgenics. All right, we can't generate transgenic people. That would be nuts. So we don't do that. Okay. All right. So horses, would that make a good model genetic organism? Why or why not? What do you think? So you should be thinking no, even if you love horses. I'm not a horse guy. I'm scared of them. But I'm not scared of them. I had a horse once, and he kicked my brother. A horse and kicked my brother, and ever since then I kicked him right in the knee. I've been very anti-horse. But anyways, poor genetic organism, model genetic organism. First of all, they're real big, right? It's going to be real expensive to feed them, keep them going. They don't have a lot of progeny, right? They only have one. I've never heard of twin horses. I wonder if that you can have that. I'm sure you can. Really short, really long generation time, right? You're going to have to wait years for that thing to grow up and develop. So, yeah, too expensive to house feed and propagate, too few progeny, and too long of a generation time. Okay, so a little bit on the history of, not necessarily the history of genetics, well, the history of genetics, but like the thought of genetics. Genetics has been going on for a long time. Early man, without knowing so, really, he was doing genetics. I mean, early on farming is a really example of genetics, domesticating plants and animals. And even the ancient Greeks had theories on like pollination and generating um, hybrids, hybrid organisms, the mule. Um, Example of making wheat like with really large seeds so that it doesn't blow away and stuff like that. Yeah, modern wheat, um, more numerous and larger seeds. They won't scatter before the harvest. You can collect them very easily. And even this Babylonian artifact, right, was depicting individuals pollinating date palm trees. All right, so what were some early and incorrect thoughts? And I'd like to see how the thought, modern genetics, what we know today about DNA and genes. People had no idea about DNA back then, right? So how did they think we got our genes? Well, one of the earliest fields of thought was something called pangenesis. Don't confuse that with Pangea, right? That's the big continent. Pangenesis. And this carried on for a really long time. Look at this guy. Darwin was even a proponent of pangenesis. And in pangenesis, the thought is that every part of your body has its own genetics. And then, like um, before the fertility particles are made, right? The sperm and the egg. Those genes would transfer from all of those different parts to the gametes, right? These particles, they called them gemmules, and they would carry information like from your arm or your fingers or your eyes to the reproductive organs, and then that would pass to the embryo at the moment of conception. This was pangenesis, and obviously that's not true, right? So there was the guy that did the brilliant experiment, really simple, brilliant experiment, disproved it. What he did is he took mice and he cut their tails off, and then he mated them, and he kept cutting their tails off after generation. I really want to look up the paper to see how many generations he kept doing this. And the mouse still grew a tail the same size. But pangenesis would say that over time, over generations, the mouse would eventually lose that tail, and that's not true. So here you can see the genetic information is traveling from all different parts: the heart, the brain, the ankle, right, to the sex organs, and then being passed on to the sperm and the egg. Whereas the correct theory, germplasm theory, which is what we have today, everything already lies there in the gametes. The sperm and the egg already have the copy that they need of the genome. That's not a full copy, right? And then that gets passed on during fertilization. Okay. All right. So, early on, inheritance of acquired characteristics, right? So this is a field of thought. And this is like you acquire traits in a person's lifetime, right? So maybe, I don't know if this is the best example, but like a giraffe neck getting longer, right? So as you grow and you need food higher up in the tree, your neck gets longer. A good example would be musical proficiency, right? So throughout life, you get better at playing the dulcimer. And so that means that your son is going to be top-notch dulcimer player, right? Well, I think a lot of this stuff was justifying, politics probably, and monarchs staying in power. But obviously, this isn't true, right? If you become an expert violinist, that doesn't mean your kids are going to be good at violin, unless you're Deion Sanders, right? If you're Deion Sanders, then your son, sorry. Okay, another thing is preformationism. Preformationism, this is my favorite. So with preformationism, inside the egg or the sperm, I guess they didn't figure out which one, you have what's called a homunculus. And a homunculus is basically a tiny little person. And, and all your traits come from one parent. So this train of thought lasted, through the 1700s. Pretty crazy, right? We all come from a homunculus. And, maybe a lot of this was tied to religion. Who knows? I don't know. I'm sure that information is out there. Very interesting. But there's what you look like. You're all shriveled up. You got a giant head, and you're crouched down, and you're a tiny person. All right, now we're getting maybe a little more reasonable. So blending inheritance, but it's not true. Genes do not blend. But a lot of these things, like the blending, a lot of these characters were seen in some experiments performed by famous geneticists. So Gregor Mendel, father of genetics, who did his experiments with the pea plant. We're going to talk about him here for the next 2 units, and we're going to keep talking about him throughout the whole semester. He keeps coming up. That's why you guys really need to get this first few units down. Be sure you're working really hard these first few units. So he did see this, but it's not an actual blending, and we're going to see, at the molecular level, really what happens when, a white flower and a purple flower, or a white and a red flower, are mated together and you get a pink flower, right? We're going to see why that actually happens. It's not blending. Let's talk a little bit about Gregor Mendel here. We're going to talk about him a lot. He's probably my favorite biologist. Amazingly, this guy was a Franciscan monk, and look what he did to the church. He really went, and he used church funds to provide for his research. He had the monks in his abbey counting his peas and stuff. But he discovered the basic principles of heredity. And what made his work so powerful, though, was the fact that he used mathematics. He applied statistics, which is in many forms undeniable proof that his concepts were correct. And he's now known as the father, generally recognized as the father of genetics. The other big guy would be Thomas Hunt Morgan, who worked for Drosophila, who also did some amazing discoveries. But this guy, he went from homunculus to all these theories of heredity that were pretty amazingly applied. And I think as we get through them, without any knowledge of DNA or mitosis and especially meiosis, the fact that he was able to figure this stuff out is pretty remarkable. So Schleiden and Schwann gave rise to what's called the cell theory, where the term Schwann cell comes from. And the cell theory states that all life comes from cells. All life is composed of cells, right? So the fact that the mouse and a blue whale have similar size cells, right? There's other reasons for that, but it's all part of cell theory. And also that cells arise from pre-existing cells, and of course that a cell is the fundamental unit, the smallest living unit of structure and function in living organisms. Walter Fleming discovered chromosomes. He observed them under the microscope in the late 19th century. And he published description of mitosis. So mitosis and meiosis are crucial to genetics. And we're going to study that here in chapter 2. It's really important that you fully understand what is happening during meiosis. And we're going to drill it into you here. But, this led to the acceptance that it was ultimately the nucleus that contained the hereditary material. Charles Darwin, evolution, right? This is more relevant to population genetics, but survival of the fittest. And he published his ideas in that famous paper, The Origin of Species, in the mid-19th century. August Weismann, this is the guy that did the mouse tail experiment. 22 generations. So he cut mouse tails over all these successive generations, and the mouse just kept growing. Mice kept growing tails. Right? So it disproved that theory of pangenesis and reinforced the germplasm theory, which is widely accepted today. Walter Sutton discovered the gene, right? So that gene, he didn't make the term gene, but he discovered that genes were located on chromosomes. And then, of course, Thomas Hunt Morgan really began to unravel a lot of the things that were brought up by Gregor Mendel with his use of the model organism, the fruit fly, and in transmission genetics. All right, modern genetics, though, we've got a lot of fancy equipment and techniques that are applicable, and we're going to talk about these in this course as well. There's a whole couple chapters. Really important that you get these techniques down and understand them, how they work. And if you're taking the lab, we're going to do some of these techniques as well, including sequencing DNA. We're not going to do that in the lab. DNA amplification, which is PCR. We're going to do that. And things like that. All right, almost at the end here. Important terms, eukaryotic versus prokaryotic. So what do these terms mean? Well, basically, it's a type of cell. It describes a cell. And prokaryotic are bacteria, and eukaryotic are everything else, right? They include animal cells, plant cells, fungal cells, the paramecium and the amoeba. But prokaryotes, all in their separate cellular division. What is a gene? It is the fundamental unit of heredity, right? So collectively, the genes in the genome define our genetics. And genes come in multiple forms. Now, usually, they come in 2 forms. And that's because many model organisms have only 2 copies of the genome. This is what we're going to start to get into in the next chapter. And each of those copies are called an allele. So if you have 2 copies of chromosome number 3, and you have the gene for blue eyes on both chromosomes, right? Well, you could have the gene for, if you have the gene for eye color on both chromosomes, on one chromosome, you can have the gene for blue eyes, and on the other chromosome, you can have the gene for green eyes. Okay, so those forms of those genes are called alleles. Very important term, right? These are genetic variants, right? And they exist on different chromosomes. And then, so you have the gene, which is the DNA code, and then you have the phenotype, which is the observable characteristic, right? So, you got a gene for blood pressure, right? And that's not true. Obviously, a lot of genes comport to blood pressure. It's just a silly example. But you can detect blood pressure, right? It's not just something you see. You can visibly see. It's anything you can detect. Even at a molecular level, maybe you can detect it. But genotype confers phenotype. And then DNA and RNA, deoxyribonucleic acid and ribonucleic acid, right? This is, where the genetic information is found. Right? Ultimately, it's on the DNA. That gene is transcribed into RNA, which is then translated into protein. And then these genes are located on chromosomes. Chromosomes separate through mitosis and meiosis, right? And that is really, when we talk about mitosis and meiosis, I don't care. We don't care about the prophase, metaphase, anaphase stuff. That's not what's important anymore. What we're looking at is that segregation of chromosomes, right? That prophase, metaphase stuff, done with that in Bio 1. But now we want to really look closely at how the chromosomes separate, what sort of processes occur when those chromosomes separate, before they separate, the flow of genetic information, how it goes from DNA to RNA to protein. That's going to be in the second half of the course when we talk about molecular genetics. And also, how these different alleles arise, right? Well, mutation. Is mutation a bad thing? Not always. If it makes you more fit for your environment, that's a good thing for evolution, a good thing for your species. Maybe it's enough mutation to change you into another species. You never know. But these are permanent changes in the genetic information, in the DNA, and they get passed from one cell to another cell. Now, if you're doing mitosis, it's from one cell to the daughter cell. But if you're doing meiosis, if the mutation is in the germline, then that permanent change gets passed from parent to offspring. Genetics is not simple, though, and we're going to see examples of this starting to pop up here in chapter 4. Right? That's when most of the examples pop up. But many traits are affected by multiple things, multiple genes, multiple factors, environmental factors, health factors, age factors. And then also the term evolution. This is like genetic change over time. Very important distinction, because I've come up with some, I've thrown a lot of terms out here, and these are terms that some people get confused, and it is absolutely essential that you fully understand and be able to reference these terms correctly in this course. That's trait, characteristic, allele, and phenotype. So, if you have the gene for eye color, right, you have alleles for specific genes, like blue eyes and green eyes. Well, we would say that eye color, which is more general, would be a characteristic, right? So characteristic is kind of synonymous to gene, whereas trait is synonymous to allele. Okay, so trait is a specific variant of a characteristic. An allele is a specific variant of a gene. Now, phenotype covers both of these terms, trait and characteristic. But I would say it does not cover allele and gene, right? So, but it does cover trait and characteristic because it's an observable feature. So you can observe blue eye color. But if you're talking about certain alleles that you can't visually observe, we would not necessarily use the word trait, because it cannot be visibly observed. But we can still say it's a phenotype. So that's what I mean by they're somewhat synonymous, right? Trait and allele are somewhat synonymous, but not completely, right? But very important that you understand that distinction between trait and characteristic. Like, trait is specific version, characteristic is more general, but phenotype covers both of these. Okay. All right, that wraps up chapter 1 here. So you guys will have this going out the door here on Monday. And I'm going to try to get chapter 2 done here today. And I got to get some more coffee. Hope you guys are having a great first week back. And I'm really looking forward to working with each of you in this course. Open door policy. Email me. I'm pretty good about response time. If a day goes by and you haven't heard back, like a full day, the next day, email me again. I just probably got too bottlenecked with my emails, and I just haven't had a chance to get back to you. But don't feel crazy about doing that. That's okay. It doesn't happen often, but I'll miss emails occasionally. So I'm not out to get you. I just got overwhelmed, and please email me again. It's no problem. Okay. Trying to think, that textbook, if you haven't get going on Achieve. All right. This course should all be set up through Barnes and Noble first day. So, you're going to need the online resources, obviously, because otherwise you're not going to be able to do the assignments and your grade is going to suffer. And you are going to need the book. You cannot get around getting another edition or anything like that. Okay, so just stay opted in, don't opt out. And also if you have problems with anything in Achieve, like if you have technical problems, understand that I'm only limited. There's only so much I can do to help you. Because I'm not a computer guy, as you can see. I probably shouldn't even be using Microsoft Teams. I think I should be using PowerPoint dictation or something. I don't know. But I'm not a computer guy. So you may need to contact technical support, but I haven't had too many issues with this in the past. Usually everything works pretty nice, but you may need to contact their technical support if you're having problems. Also, 9 times out of 10, though, clearing your cache, your search history, your browsing history, restarting your computer will fix the problem. Using a different browser, try all those things first before you reach out to me or them. And just don't be disappointed if I tell you, I'm sorry, I have to send you to Macmillan to assist you with your problem. Okay? Anyways, I'm out, and I will see you for Chapter 2.